Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore the types of gene mutations. We will discover how microscopic changes in our DNA sequence can have profound effects on living organisms, driving both evolution and genetic diseases. Let us dive in and see how this works. To start our journey, imagine you are baking a cake using a very old, famous recipe book passed down through generations. If you accidentally copy the recipe and write the word salt instead of sugar, the final cake will taste completely different, even though you only changed one single word. In biology, your DNA is that ultimate recipe book, and the letters of the DNA alphabet are used to build everything in your body. Sometimes when cells copy this recipe book, they make a tiny typo, like swapping one letter for another or accidentally skipping a word entirely. These typos in the DNA sequence are what we call gene mutations. While the word might sound scary, it is simply a natural copying error that happens in all living cells. Now that we have an intuitive understanding, let us look at the formal definition. A gene mutation is formally defined as a permanent, heritable change in the nucleotide sequence of a specific gene within an organism's DNA. To truly understand this, we must look at the specific types of changes that can occur at the molecular level. The most common type is a point mutation, which involves the alteration of a single base pair in the DNA double helix. Point mutations often manifest as substitutions, where one base is simply replaced by another. This is the molecular equivalent of swapping a single letter in our recipe book. Building on the idea of substitutions, we can categorize them further based on the chemical nature of the bases involved. DNA bases are divided into purines and pyrimidines. When a substitution occurs, it can be a transition, which is when a purine replaces another purine, or a pyrimidine replaces another pyrimidine. Alternatively, it can be a transversion, where a purine replaces a pyrimidine, or vice versa. These specific chemical swaps change the genetic instructions, and understanding them helps geneticists trace evolutionary lineages and identify the exact nature of genetic disorders. Beyond simple substitutions, we have another major category of mutations that are often much more disruptive. These include insertions and deletions, where extra base pairs are added to or existing ones are removed from the DNA sequence. Because the genetic code is read by the cell in triplets, called codons, adding or removing a base is like adding or removing a letter in a sentence of three-letter words. It shifts the entire reading frame. This is known as a frame shift mutation. A frame shift completely alters the reading frame and changes every single amino acid downstream of the mutation site, usually resulting in a completely non-functional protein. Understanding these different types of mutations leads us to ask why they matter so much in the grand scheme of biology. Gene mutations are incredibly important because they are the ultimate source of all new genetic variation in the biological world. Without these tiny changes in our DNA, evolution would be completely impossible, and all living things would remain exactly the same forever. In the real world, mutations are responsible for the beautiful diversity we see in human eye color, hair texture, and height. However, they also have serious medical implications, as specific mutations can lead to genetic disorders like sickle cell anemia, cystic fibrosis, and various forms of cancer. Understanding the exact type of mutation a patient has allows doctors and scientists to design targeted gene therapies and personalized medicines to treat these conditions at their root cause. To study how often these genetic typos occur in a population, Geneticists calculate something called the mutation rate. Conceptually, this gives us a mathematical way to measure the frequency of new mutations arising in a specific gene per generation. Understanding this rate is crucial for predicting how quickly a genetic disease might spread or how fast a species can adapt to a new environment. We represent this concept using a simple but powerful algebraic formula. In this formula, the Greek letter mu represents the mutation rate per gene per generation. The variable m stands for the total number of new mutant alleles observed in the offspring of a population. The variable n represents the total number of diploid individuals in the population. If the number of new mutations, m, increases, the mutation rate will naturally increase. Conversely, a massive population size n with very few mutations will result in a very small mutation rate. Now that we have our formula, let us apply it to a real-world scenario. Imagine a geneticist is studying a large population of exactly 100,000 mice to track a specific gene that controls fur color. In the newest generation, they carefully examine the offspring and discover that 4 mice have a brand new dominant mutation for white fur. Neither parent had this trait, so these are entirely new mutations. We are given n equals 100,000 and m equals 4. Because mice are diploid, we first find the total number of alleles by multiplying n by 2, giving us 200,000. We then divide our 4 mutant alleles by this total allele count. This gives us a final mutation rate of 0.00002 mutations per gene per generation. Physically, this means that for every 100,000 alleles passed to the next generation, exactly 2 will carry this new mutation. Having successfully calculated the mutation rate, let us take a moment to prove why the denominator of our formula is exactly 2n. What we are establishing here is the logical derivation of the total genetic pool. The mutation rate is fundamentally a probability, representing the fraction of newly mutated alleles out of all the alleles passed on to the next generation. To find this fraction, we must divide the new mutations by the total number of possible mutation sites. Since we are dealing with diploid organisms like mice or humans, every single individual carries exactly 2 copies of the gene in question. Therefore, the total number of alleles in the entire population is 2 times the number of individuals, n. This mathematical reasoning perfectly explains why our formula uses 2n in the denominator, ensuring we account for the entire pool of genetic material where a mutation could potentially occur. Moving from population genetics back to molecular biology, let us look at how we calculate the physical impact of a frame shift mutation on a protein. Conceptually, when a deletion or insertion occurs, the cellular machinery reads a certain number of normal amino acids before hitting the mutation. After the mutation, it reads scrambled amino acids until it accidentally hits a new, premature stop codon. We can represent this with a simple formula. The total length of the mutant protein, L mutant, is equal to the number of normal amino acids, N normal, plus the number of scrambled amino acids. N normal represents the building blocks that are identical to the healthy protein. If the mutation happens early in the sequence, N normal decreases, and the resulting protein will be severely shortened and likely non-functional. Let us apply this logic to a specific example. We are given a normal gene that produces a functional protein exactly 150 amino acids long. A deletion mutation occurs at position 30 in the sequence of codons. This frame shift creates a premature stop codon exactly 20 codons downstream from the mutation site. We need to calculate the length of the new mutated protein. Because the error occurs at the 30th position, The first 29 codons are read perfectly normally, so N normal is 29. The cellular machinery then translates 20 scrambled amino acids before hitting the accidental stop codon. Adding 29 and 20 gives us a final answer of 49 amino acids in total. Physically, this means the cell produces a severely shortened protein of 49 amino acids instead of the normal 150, which will almost certainly lead to a severe genetic disorder. To solidify our understanding, let us look at this diagram illustrating the different types of gene mutations we have discussed. At the top, you see the normal gene sequence, neatly divided into 3-letter codons, each translating into a specific amino acid, ending with a stop codon. Below that, on the left, we see a substitution mutation. Notice how a single base is swapped, highlighted in red, which changes only one amino acid, from glutamate to valine. On the right, we see an insertion mutation, where an extra base, highlighted in green, is added. Notice how this shifts every single letter to the right, completely changing the amino acid sequence from that point onward. Finally, at the bottom, a deletion mutation removes a base, marked with a red X, causing a similar frame shift. This visual perfectly demonstrates why frame shifts are so much more destructive than simple substitutions. We have covered a lot of ground today, so let us summarize our key takeaways. First, we learned that gene mutations are fundamental, permanent changes in the DNA sequence that act as the raw material for evolution and genetic diversity. We explored how simple point mutations, like substitutions, can alter single amino acids, while insertions and deletions cause highly disruptive frame shift mutations that change the entire reading frame of a cell's genetic instructions. We also learned how to calculate mutation rates, which allows geneticists to track and understand how genetic diseases arise and how populations adapt over time. By understanding these microscopic copying errors, we gain profound insights into the diversity of life and the foundations of modern medicine. Great job following along, and keep exploring the wonders of genetics.